So I'm very happy. My name is Aurora Clark, and I'm at the University of Utah. And today I am moderating Professor Elizabeth Munch's talk, who is an associate professor now. Congratulations. She recently got tenure of computational mathematics, science, and engineering at Michigan State. And she's going to be talking about directional transforms, which sounds like I actually really love your title, about how to look at data from every direction at once.
All right, thank you. And thank you all for the invitation. Can you guys see my slides? Is this good?
Yeah, it looks great.
All right. So thank you very much for the invitation. I was, when I was trying to come up with the title for this talk, this was, I had a bit too much fun making the title slide, but this is, I wanted to talk about an idea that I think is really interesting coming out of the TDA community, and I think has a lot of potential for really interesting applications for a whole lot of reasons that I'll get into. But the sort of general idea is that we're going to study shape data using this idea called a directional transform. So the talk I'm going to give is not going to be chemistry. So sorry, it's going to be, I'm going to use this idea on some applications from plant biology where we can sort of talk about some specific use cases. But at the very least, I'm hoping that we can sort of start the discussion about where you could possibly use this in some sort of chemical type application. So to that end, okay, so I know there's a lot of AATRN people around, and so this is kind of the like preaching to the choir, but this is sort of the general way that I tend to think about data and data science, which is not usually what people think of when they think of data science. So I like to think about data in terms of shape and structure. And in a lot of cases, the shape and structure is sort of the obvious naked eye shape and structure I might want to think about. This is one of these talks where I get to not try to convince you that there are other weird ways to get shape and structure out of your data. But in this case, I'm actually going to talk about straight up shapes that you can see. And so a lot of the applications that I've been interested in are things like X-ray CT scans of plants. So in this particular, let's see, can you guys see my mouse, I think? So for instance, I have my scans of walnuts over here. I've got an aloe plant. I've got scans of tree trunks where you can see the inner workings and the rings. I've got, this is a red bell pepper down here. This is some form of citrus. There's a lot of really interesting cases where we can get these data sets where you have interesting shape and structure, and you might have a lot of these particular data points, right? So now a data point is, you know, a walnut or something. And I want to be able to say things about the shape and structure in some sort of automated fashion. And as a not biologist, I want to do this where I can make my computer do it, because I don't want to have, you know, undergrads sitting there pointing and clicking for 300 hours, like finding all the endpoints of these walnuts or whatever. I want to have something that's a bit more automated. So to that end, that's what we're going to talk about today. So a lot of the pipelines in TDA, in topological data analysis, tend to fall into the following sort of framework. You have some sort of input data set, which again, as a mathematician, data scientist, I get to be super general about that, but it's whatever my domain scientist collaborator happens to be interested in at the time. The usual plan is to try to figure out a way to turn that data point into some sort of topological signature. So some representation of the shape and structure in a way that encodes something hopefully meaningful about the question that I'm trying to ask. And depending on the day, this can be a lot of different things. And then once I have this topological signature information, I want to figure out a way to do analysis on this, which means I need to be able to do things like machine learning and statistics, and really be able to take these weird topological inputs as some sort of input to something that is a reasonable machine learning or statistical method. So in general, the way I tend to think about these methods is we're trying to find ways to summarize shape and data with a lot of goals in mind at the same time, right? So quantifiable, just meaning that I have some sort of quantitative object I can work with no matter how weird it is. Concise, meaning it's smaller than the original data set. Comparable means I have access to things like metrics, and robust, meaning under those metrics, these sort of representations are nice, even if you're given sort of noisy variants of it. For the purposes of today, we're going to talk mostly about the concise bit, because we're going to have a sort of interesting way of taking your particular data set and encoding the shape in some manner that I can use for other sorts of inputs. There are a lot of types of topological signatures. There's lots of things that this sort of field has been using as inputs. I'm actually going to talk about the least complicated version that's on this slide. So the stuff I'm going to talk about today is going to be entirely focused on thinking about Euler characteristic curves. And so we'll go into what that is in a moment. But this is sort of a special case, or maybe a summarization of the information in a persistence diagram, if you've seen something like that. And it's going to be much easier to compute and much less complicated than some of the graph-based signatures that are also very interesting for this particular field. Right, so just as sort of a general mantra of where we're going to go with this, I like to think about getting these applications, and one of the main things I'm trying to do is to find a way that I can encode information in whatever my starting data set is in a way that's useful for the domain scientist, right? So a lot of times there's a bit of an ad hocness, but also an art to figuring out, if you have this particular input data set and you understand, if you eventually can talk to the biologists or whoever about the understanding enough of the question of what's going on, can you figure out the right or a good way of encoding something about your shape and structure? But also do this in a way that respects the topological information from your starting input data, but also can be used for analysis, and in particular is computationally feasible when you have huge numbers of these things. So for this talk, we're going to focus on the Euler characteristic curve and the Euler characteristic transform, and in particular, I'm going to talk about an application of this to barley morphology that we worked on that was very successful. Okay. So Euler characteristic transform. So what is this thing? All right, so let's go back to whatever, 1800 something. So Euler is bored, and like Euler does when Euler is bored, he comes up with brilliant mathematics. So one of the things that Euler noticed was that if you take any sort of standard solid, cubes, tetrahedrons, octahedrons, anything like this, you can look at the number of vertices, the number of edges, and the number of faces. All right, so in this case, vertices, I just mean the corners of whatever this thing is. Edges, I mean the edges, the bent parts that go between them, and then faces are sort of any sort of flat bit that went on the sides. So in the case of the tetrahedron, I have 4 vertices, 6 edges, and 4 faces. If I sit there and count, and I'm going to trust me writing this earlier. And what Euler noticed was that if you take that those counts of all of these objects, if I take the number of vertices minus the number of edges plus the number of faces, I get 2. And it turns out if I do this for any platonic solid, or really anything that sort of looks like this sort of standard, maybe something that a ball could be squashed into space, the answer was always 2, which is weird, right? So there's no reason for this to make any sense. It doesn't matter if I took my octahedron and broke it up into more pieces and had additional vertices and edges and faces and things like that, I would still get 2. So it turns out that this is an example of what's called a topological invariant. So the idea with topological invariance is that I should be able to take some sort of topological space and spit out some representation of it that doesn't change up to homeomorphisms of the space. So homeomorphism means I get to stretch it, pull it, do anything except for rip it or glue it back together. And if I do any sort of breakup of this space, if I break it up into vertices, edges, faces in any sort of possible configuration you can think of, no matter how I do it, I'll always get the same Euler characteristic out, right? So it turns out that all of the examples I showed you on the previous slide were all things that were topologically equivalent to spheres. And so it doesn't matter what I do or how I break up this thing that kind of looks like a weird sphere, I will always get an Euler characteristic of 2, right? On the other hand, I can do this for other sorts of spaces. So I can do things like anything that looks like a disk has an Euler characteristic of 1, anything that looks like a circle has an Euler characteristic of 0, an interval has an Euler characteristic of 1, and I can basically have this information for any sort of space you could want, right? So the thing that I want to point out is that different Euler characteristics mean if you give me a space or give me 2 spaces and you compute their Euler characteristics and they are different, then I can promise you that those 2 spaces are different. However, if you give me 2 spaces and they have the same Euler characteristic, then I can't actually tell you anything. And so we're kind of stuck, right? So the other thing to point out here, though, is that this is also a very reductive version of representing shape, right? I have taken this complicated mathematical object and I have turned it into a number, and that's decently unreasonable. So let's talk about how we can use that for data, right? So In my case, I'm going to think about my input data being 2D or 3D scans. And for the pictures for the next couple of slides, it's going to be 2D just because it's easier for me to draw stuff. But you could actually do this in higher dimensions, no problem. I'm going to assume that I've got some sort of thresholded image. So at this point, whatever my original scan was, I now just have a 0, 1 matrix that's telling me whether or not something is part of the shape I'm trying to study or not. And if you hand me this binarized image, so I can kind of think about a binarized image as being some sort of matrix with, I've got vertices and edges and faces, and I can, sort of fill in pixels or not. I can then count how many pixels are part of this particular object. I can count how many vertices there are. I can count how many edges there are, and I can compute for you some sort of Euler characteristic. So in this case, if I just handed you this, I believe this was a fern leaf scan, I can calculate its Euler characteristic, and I can tell you that it's -200. Great, what do I do with that, right? So this is not quite enough for me to do anything particularly useful because that doesn't tell me a huge amount of what's going on with this very complicated picture in front of you. So here's what we're going to do to try to mess around with this Euler characteristic idea. What I'm going to do is I'm going to, again, start with some sort of binarized image, region and Rn, however you want to think about this. And I'm going to pick a direction. So in my particular example right now, my direction is following that blue arrow down that way. And the idea here is that I have a way of coming up with a function that basically follows that direction. If you like to think about it this way, this is essentially just taking an inner product with of the data point location that you're interested in with the direction you happen to be pointing in. If you want to think about it in a more graphically visual sense, right, it's level sets of this function are essentially, I guess I'm increasing going this way, right? And so as I keep going that way perpendicular to this particular line, I have increasing values of this function. All right, so now I've got a binarized image and I've got some sort of function defined on it. And if you've hung around TDA people, then you know that the next thing you want to do is start taking sublevel sets, right? So fix a direction, define this function, and then we're going to look at sublevel sets. And so the sublevel set idea here, so in this particular picture down here, my direction is going this way. So for any sort of function value I'm interested in, I'm looking at the portion of the picture that is to the left of that particular line. And what I can do is I can look at the Euler characteristic of the portion of the picture that's to below that line, or left, or whichever way I'm coming from, right? So what I can do is for a bunch of different threshold choices, I can figure out what this Euler characteristic is. And so this value is probably, I don't know, somewhere in, oops, no, behave yourself. Stop. Oh, never try to do technology in public. All right. This value is probably something along the lines of one of these values down here. And so I have an Euler characteristic value for that particular choice of threshold. And as we are wont to do, I'm just going to mess around with increasing that threshold and try to see how the Euler characteristic changes, right? So in this particular choice of direction, I have this Euler characteristic curve, which is the portion of it that's off here to the left. But then I can do this in a different choice of direction. So here's another direction, which I think is probably going in an up leftish direction. And I get the Euler characteristic curve that's in the middle here. And I can do this in another direction. I get the Euler characteristic curve that's over here. And so I can do this in a whole bunch of different directions. And so there's no real immediate reason for why I should want to do one over the other. Okay. If you don't have a good reason to do one over the other, again, this is going back to our standard TDA mantra, you might as well just do all of them at once, right? So what I'm going to do is I'm going to take my Euler characteristic curve. Sorry, I should have said that. So Euler characteristic curve is what I mean by pick a direction and get this function out of it. Euler characteristic transform is now take those functions in each of these directions and sort of stack them up together and try to see what's going on. So in my particular case, because all my pictures back there were in 2D, I get a direction for everything on the circle, so everything in S1. And any direction in S1 then defines this Euler characteristic curve, which is really just a function that takes in real numbered threshold values and spits out an integer for my Euler characteristic value, right? And so what I've done is I've basically taken my fern leaf at this point and encoded something about the shape and structure of it in this matrix, which you can either think about as being a 0 to 2 pi by Euler characteristic threshold value on the side, Or we can think about this as being something like S1 cross these vectors. And so you start getting these little cylinder representations of what's going on with your particular structure. Okay, so, right. Why do I want to, what's the justification for doing all of this at once? So it turns out that this process, the Euler characteristic transform, which basically takes in a shape and spits out this giant cylinder looking thing, is actually injective on the space of simplicial complexes that you could have built in R2 or R3. And so what that means is that if you handed me two of the, let's get injective. If I have two spaces that get mapped to the same Euler characteristic transform, that means they must have been the same to begin with, right? So the upside here is that this is a lovely representation for differentiating what's going on with your particular shapes and structures, right? So unlike before, where if I had the same Euler characteristic curve, I couldn't promise you any, or the same Euler characteristic period, I couldn't promise you anything. Now I can actually take this pile of data and use it as a representation of my simplicial complex. Okay, so that's a lot of building up what it is that we're doing. So let's talk about how we can use this on something. And so we're going to focus on this application to barley. As with any super applied project, this involves a lot of people and a lot of years, and this is kind of the extreme version of it. So we got access to this barley experiment data. This barley experiment had started in the 1920s in UC, I'm going to say it wrong, and this is going on YouTube. One of the UC colleges, which I'm going to remember later, and I'm sorry, Dan Koenig, I'm not going to get it wrong on public. So what they had done was they had taken, Harry Harlan and Mary Martini, who are the people up here in the top, they had taken a bunch of trips and went and found a bunch of different barley lines from a bunch of different places. And so you can see all the different barley lines that we're going to be interested in over here on the bottom left. And so they went and they found all these different barley examples. And what they did was they cross-pollinated all of them and put all this stuff out in a field and let them just grow. The idea was this is an artificial evolution experiment where we could try to see things about what do I know about, or what can I find out about sort of genetics and traits and what gets passed on without a particular ton of control. So what they did was they put this out and they put it in the field and they let it cross-pollinate and they got more seeds, and then they basically took those and they stuck them out in the field again. And they did this for something on the order of about 50 years, all right? And they saved everything. And so what we got access to, so the current person taking care of this data set is Dan Koenig. And so he sent us, and by this I mean literally mailed from California, these barley panicles coming from this experiment that we were able to scan in my collaborator Dan Chitwood's machine and get X-ray CT scans. So what you can see here is a snapshot of a scan of one of these barley panicles that we got out of this X-ray CT machine. All right. Okay. So here's what we ended up doing. And by we, I mean all the credit goes to Erica Mezquita, who you can see in the bottom right, who did all of this work. So what we did was we got these X-ray CTs and we messed around with a lot of ideas for what to do with this particular data set. And what we ended up doing was actually segmenting out all the seeds to study their shape. So Eric's first job was to take these scans of panicles and segment out all of these seeds from the panicle. So we are not removing them physically from the panicle. He's doing this entirely on the computer. And we ended up with something on the order of about 38,000 seeds. All right. So what we were trying to do was to understand things about the relationship between the seed shape and the different information and strains in the genetics of where these things came from. All right, so we have sort of the traditional descriptors, which are already kind of nice with these scans because you can automate things like length, width, height, volume, all sorts of other things. And then we also built into our system ways of computing the Euler characteristic transform for each of these seeds. So in this particular example, what we did was we picked a lot of directions, 158. And so for any of these seeds, you then represent this thing as this giant pile of data corresponding to our Euler characteristic curve information in at least 158 directions. So the results were lovely. So the first thing that we did was we focused on just the original founders. So just take the original strains that were found in all these different locations and try to see if we can match something to do with the shape with the strain that this came from. All right, so in this particular case, what we ended up doing was we started by looking at 11 different traditional descriptors, and I don't remember all of them, but again, length, width, height, that kind of thing. And then we also built in the topological descriptors. So in this case, if you do topological descriptors this way, it was 158 directions times however many thresholds we did was very, very large. What we ended up doing was honestly just taking that data and mapping it down to 12 dimensions using UMAP, and that got us a much, much lower description of what we were using. So what we found was that if you try to then take your input seeds and predict something about the original founder that it came from using just the traditional descriptors, we were getting something on the order of 57% accuracy. The topological bits themselves was doing better, but actually we did absolutely the best by having a combination of both of them. So take both the traditional descriptors and the topological descriptors, and we were getting something in the 80% range for predicting these founder lines. So again, if you hang around NeurIPS people, they start to freak out because it's like not 99.99%. If you talk to biologists, this is incredibly exciting because anything that's predicting this is super, super nice. So, oh, and I know I'm probably getting close to the end of my time, aren't I? Sorry. Okay, let me try to get to the punchline so that I don't run over our next talk. Right, so one of the things that we can do with this that I think is super interesting is you can then use your machine learning model to decide which of your directions was the most useful. So we use an ANOVA test to try to figure out which of the directions of our Euler characteristic transform were actually helping us with our data. Turns out the answer is approximately what you would expect, which is great. So if you took your barley seed and you pick the directions and you line them up, it's basically the stuff, your barley seed kind of looks like a football with a thumbprint in it. It was basically the bottom and the top directions were the things that were telling us the most about it. Basically the stuff where the sort of general shape and structure of the outside is more interesting. We can also do things to try to figure out stuff about the later progeny, which is also super exciting. So remember, we trained all of our stuff on generation 0. And then what we did was we took the trained model we used on the original and used it to predict which founder we think they're most related to for seeds coming from the later generations. And so in this case, we end up having these different percentage of predicted seeds of where they come from. The reason this is exciting from the biological standpoint in particular is that so when we showed the biologists this picture, the reason they got excited was they have a lot of information about the genetics, right? So they can tell you things about how much of the genetic information from each of the different founders has made it on to the later generations. We did the same thing in terms of trying to predict the founder information for the later generations with absolutely no information about the genetics. And the lines that are the most represented in our model are also the lines that they see genetically. So right now our collaborators are off doing all sorts of fun things, doing genetic tests to try to confirm this. But basically there's these 3 strains, the Mason Carey, the CA Mario, and the Atlas that show up in a lot of, show up overrepresented here. But again, we're only using shape data to predict this. This has nothing to do with any of the genetics. Right, okay, so sorry, let me say a half a sentence about things that we are trying to do with this. There's a lot of fun games you can play with this particular setup, including things like trying to take, excuse me, directional transforms and input other sorts of topological signatures. So again, if you're an AATRN person, you can swap out your Euler characteristic curve with things like merge trees, and the computation gets worse, but hopefully the representation gets better. You can do fun things with that. And there's also a lot of really nice things that you can do in terms of using this idea where we're trying to represent other sorts of one parameterized families of whatever your data points are. So we've been doing a lot of things like this with dynamical systems and things like that, which may or may not be helpful. So with that, I think I will call it so that we have enough time for our second talk. But thank you.
Excellent. Thank you so much, Liz. That was fantastic. I think you are actually perfectly on time. We've got even maybe 1 minute for quick questions. If anyone wants to put anything in the chat.
Yeah, we actually have 6 minutes for questions. She
Oh,
didn't
okay.
use her
so I
full
timed that wrong in my head. Sorry.
Yes, so
So you had
go
25,
ahead.
yeah, we started at 7:50 by my count. So
Ah.
you were right at like 24 minutes. But if we've got 5 minutes of
Got it.
questions, then we've got plenty of time. I'll actually lead off the questions. I'm kind of curious. So have you done any kind of sensitivity analysis associated with this, right? So, I mean, obviously you demonstrated that the directional transform can differentiate between different spaces when they would have like the same Euler characteristic, right? But what about, I guess, and maybe this is an ill-formed question, but how different do they need to be before you actually get to see this differentiation in the directional transform?
That's a fabulous question. So I can give you the punting math answer first, which is that if you can tell me the Euler characteristic curve in every possible direction, and I mean that in the mathematical every possible direction, then I can promise you they're different. That doesn't necessarily tell me that in computational settings that that's good enough for what I'm doing. So we haven't actually done a full sensitivity analysis. That would be a really interesting direction to go with this. The main things that we can do is to say things like I know there's, okay, so let me also back up. There's a lot of people that are doing various versions of a slightly different question from what you're asking, which is things like, how many directions do I actually need to promise you that I can completely distinguish these objects?
Yeah.
And the answer is usually super nasty, and it's very much dependent on your particular input data. And so there's a lot of like very specific things you have to do to make sure that that happens. We've been going, at least from the application route, we've been going let's just do it in a lot of directions and see what happens, right? And by a lot, I mean, so I do think in the paper we have at least one figure that's showing things like how many of these directions, do you need before anything is kind of giving us reasonable results? And so a sensitivity analysis in that case, again, for this particular data set, we've sort of thought about that. But I think a lot of it's going to have to do with your specific data set and just making sure that you have enough directions. And also, I think the harder thing to figure out is enough directions in the directions that count. And so that's not necessarily something you know in advance, right? So if I'm handed a new data set, with the little nice little football looking things, right? I could have guessed that was probably the directions we needed, right? But I think there would be something interesting to be done if you have sort of a new data set and you're trying to figure out how many of these directions do you actually need to try to figure anything out?
So could you formalize that as an optimization problem?
Could you formalize that as an optimization problem? At the very least, I'm sure any of your favorite machine learning hyperparameter pickers could be something that's done for this. The big pro, and actually something I don't think I mentioned, but I should have, the big pro here, and this is as somebody who wrote a PhD thesis doing persistent homology, you never have to compute persistence, and it's so wonderful, right? So like computationally, this is just so much better. And so if you can do anything like this, I mean, even like the hyperparameter stuff, right? You can compute a lot of these things a lot faster because all I need to do is have counts of how many things I have at any threshold. I don't have to touch a matrix ever, which is my happy place, right? So, so I think there's some interesting things you could do.
Yeah. Excellent. Wonderful. Other questions for Liz? Oh, Leonardo's raised a hand. Go ahead, Leonardo.
Hi, thank you for the talk. It was excellent. For me, it's just a simple question. So when you first introduced the Euler characteristic, you were dealing with shapes and forms of polygons, and the topological invariants were integer numbers. But when you go to the Euler characteristic curve, it actually got like it for me, my point of view, it changed for integer number to a continuous number. Is that so, or
nope,
I'm
it's
missing
actually
something?
an integer valued function. Yeah, and sorry, I'm drawing lines between each of these edges, which somehow pretends that there's some sort of continuity between them, but no, it's actually an integer valued function. So each of these values are going to be fixed at a bunch of integers, and that's sort of your starting input data.
Okay.
And we have one last question from Mohammed. Do you want to speak up, Mohammed?
Yes, thank you.
Go ahead.
Thank you for the talk. My naive question is, you made mention your input data is 2D or 3D image. So my question is, can your method work with other type of data sets like the time series data that is not in the form of 2D or 3D image? Thank you.
A sort of a qualified yes, where you'd have to do something sort of fancy in terms of what you're thinking about. So for instance, if you're thinking about time series data, right? So I like to think about time series coming from dynamical systems and then spending some time trying to reconstruct your attractor and getting some sort of point cloud embedding that says something about what your original attractor was. Once you get yourself embedded into some higher dimensional space, you could absolutely use this as some sort of representation of your data, but you'd have to do some pre-processing to make it interesting. If I just have a point cloud that's embedded in multiple dimensions, right, and I take a directional transform, suddenly I just have a bunch of points and nobody cares, right? And so you might do something like, let's take the, I don't know, the RIPS complex of my embedded point cloud and try to see if I can do a directional transform on that kind of thing. The specific thing that I'm talking about here applies when you have some sort of shape and structure embedded in 2 or 3D or high, like technically you could do this in higher dimensions as well. But depending on your input data, you'd have to do something probably a little funkier if you didn't just start with a binarized image, right?
All right, thank you. And I think a kind of follow-up question, that is somewhere you made mention that you use S1 because you use 2D image. I think on the one of the function, you said you use the cycle S1.
Oh, sorry, this one?
I think yeah.
Yeah.
You said because your data is 2-dimensional, you use S1. What if it is 3-dimensional? What?
I use S2. So I try to avoid saying this in front of non-mathematicians because the counting system just doesn't make sense to me ever. But so if I am sitting in 2D data, then I'm working with directions in S1, which is really just a circle, right? So just pick a direction. If I'm working with 3D data, I'm working with S2, which is a sphere, which means pick every direction on a sphere. And so if I'm in 3D, my data here is actually, rather than being this circle cross a interval looking thing, which is a cylinder, it would be like a sphere cross an interval. And then you're working with sort of slightly more interesting structured data, but still some sort of underlying sphere cross a thing to represent something.
Awesome. Thanks so much, Liz. And I think Amir, I'm going to turn it over to you since you're in charge of our next talk. Thanks so much, Liz. Excellent. Fantastic talk. Great questions. Ask more questions in the chat if you guys want to too.